All right, so today you are going to learn a little bit about microbial genomics. And your speaker, if you weren't aware, is me. I'm not Taylor Swift, but I once managed to give an entire seminar with Taylor Swift lyrics as the titles to the slides. It took about 30 minutes before some of the students figured it out, and the faculty were still like, what's going on? So I like pop music. So who am I? Well, I'm old too, so these were popular movies when I was younger. So Austin Powers. I'm Dr. Van Laar. I'm an assistant professor in the Department of Biological Sciences here. I have a bachelor's degree in biological sciences from here. I have a concentration in genetics, which no longer exists, so I don't know if that makes me vintage or obsolete, but either way, that's what I have. And a minor in chemistry. My research advisor was Dr. Jim Youngbloom, so I did a lot of undergraduate research, and that really helped shape my academic career. I have a master's degree in biology from the University of the Pacific. My research advisor was Dr. Lisa Rishnick. I was actually just asked to give a seminar there later in the semester, so I'm pretty excited. I get to give a seminar at two of my alma maters. I have a PhD in biology with an emphasis in cell and molecular biology from the University of Texas at San Antonio, and my research advisor is Dr. Janakiram Seshu. So after that, I went ahead and did a postdoc. I was a National Research Council postdoctoral fellow. So it's a competitive fellowship. You have to write a fellowship application, like a grant proposal basically, to get one of these fellowships. I was selected and I was able to do my fellowship at the U.S. Army Institute for Surgical Research in the Dental and Trauma Research Detachment. And they affectionately call that USA ISR DTRD. So if you've ever worked with the military, it's just a chain of acronyms. My brother, luckily, was in the Navy at the time, so I could text him and be like, what are they talking about when they say this? And he would summarize it for me. It's a whole new language. And that was in San Antonio. It was at Fort Sam Houston in the Brooke Army Medical Center, which is one of the largest burn centers in the U.S., and also has what's called the Center for the Intrepid, where soldiers who were injured in combat, such that they lost limbs, could come and get prosthetics and then do physical therapy to get back up to strength. And so there, my research program focused on antibiotic-resistant biofilms and polymicrobial biofilms using samples that were isolated from patients who were soldiers injured in Iraq or Afghanistan. After that, I took a faculty position at Fresno State, so I joined the CSU system. Not quite here yet, but pretty good gig at Fresno State. I was an assistant and then associate professor of microbiology. And I guess my big career achievement, I'm nothing like Dr. Gousset. I don't have nearly as much research funding, but I was very honored to be selected as one of the provost award winners for promising new faculty. And again, I don't have the research cachet that she does, but it was mostly my teaching. I taught 17 different course preps in 5 years. So I taught a lot, but I also did have a very strong research program. I had students present posters and give oral presentations at 60 presentations at different conferences, local, national, and international. So that was really cool. And a lot of times people are surprised then when I tell them now I'm an assistant professor at Stan State. So why would I leave one faculty position for another? And I'll get to that in just a second. Faculty positions aren't easy to come by, and that was a pretty good one. And then also in my spare time, because I have a lot of spare time, I'm working on an MBA, which is really weird, I know, but I'm interested in human resources, and I'm interested in how people interact with one another, and I've been having a lot of fun doing that in my spare time. So again, that might make you go, well, you're a student at Fresno State, had a great job at Fresno State, why did you leave Fresno? Well, one, I graduated university, I became a microbiologist. Dr. Grosvenor sent me something like this once. He actually posted it on my Facebook, and I thought it was so funny, so I thought I would show you that. I graduated university for what? I'm a microbiologist, and I'm a lot bigger than you might expect. Okay, so why would I come to Stan State? Well, I told you I got my undergraduate degree here. I had such a great experience in the department that I always told myself that coming back to this department was my dream job. That was it. Like, if you ask me, like, what do you want to be when you grow up? I want to be a professor at Stan State. But they weren't hiring when I was ready to have a job. So I took a job at Fresno State, really close, right? It was awesome. I had great students, I had great colleagues, a lot of fun. And then this job opened up and I was like, yes! If I get it, that's great. If I don't, well, I already have another job, but I really want this one. And I got it. So I have a lot of great feelings about Stan State because I went to school here. But I also have a lot of good feelings because of the student population. So my mom was an undergraduate student when I was a kid. I was like 8 or 9. And she would bring me to school with her. And her professors were really cool with that. They really were supportive. And I thought, man, what a great environment for me to be in, where I could support non-traditional students. That is like the dream. I used to hang out in the library. I used to get snacks at the dining center. I probably don't need any more snacks from the dining center, but I still go get them. The other reason that I wanted to come to Stan State, these are my kids. They're, I don't know, like 16 months old, and my parents babysit them for free. So that helps too. I don't know if you know anything about childcare, but when they tell you how much that is and then you have the BOGO, it's a lot. Okay. So I'm really, really excited to be here. Even though it's only my second semester, love it. All of my colleagues are great. It's really funny to see some of them and remember, oh, I remember when I was in your class. I hope I wasn't a terrible student. I must have done okay. I'm here now, so. Okay, so what are my research interests? What's my background? I'm a microbiologist. I love working with bacteria. And so during my PhD, I worked on a bacterium called Borrelia burgdorferi, which is the causative agent of Lyme disease. We don't have a lot of Lyme disease in California. There's even less in Texas, but somehow that's why my PI wanted to study. But it is the most common arthropod-borne disease in the US. It's spread by ticks. It's terrible to grow, it's super hard to work with, but man, is it interesting. And so I published some papers on that. I went on to my postdoc. I told you we studied antibiotic-resistant bacteria, like Klebsiella pneumoniae, Staph aureus, Pseudomonas. So how did I transition then from being a microbiologist to this microbial genomics that's my job now? So especially during my postdoc, what we were looking at is what changes in gene expression did we see when we grew antibiotic-resistant bacteria, for example, in a biofilm, like community, right? And then we treated them with antibiotics. How does their gene expression change? Or when we grow two species together, how is their gene expression different from when we grow them apart? How do they interact? How are they apart? And a lot of the way that we try to answer those questions is through RNA sequencing. And so I did a lot of RNA isolation. We had it sequenced at a facility in San Antonio, but then we sent those sequencing reads to a facility on the East Coast where we had a collaborator, Dr. Chen, who did all the bioinformatic analysis. So he would send us a list of genes and say, these are the ones that are differentially expressed. I don't know what that means, but figure it out. And so then I would sit at the computer and figure out what that meant. But I really felt sort of disconnected sometimes from the data because I didn't know how we went from the sequence to the list of genes that were differentially expressed. And I wanted to know how. I wanted to feel like I was participating in every step of my data processing. And that's just me. Some people are happy to not be involved in that step. So when I got to Fresno State, I still didn't really know how to bridge that gap. I knew how to send samples to a sequencing center, But then I got in trouble because I couldn't afford to pay someone to analyze my data for me. So when I first got to Fresno State, I worked on things that I knew and things that were a little bit less complicated. So I worked with Pseudomonas aeruginosa still. I worked with, well, I didn't touch the ticks because even though I studied tick-borne diseases, the ticks creep me out. So one of my students would go and collect ticks, and then we would try to identify what bacteria were in there. And then I had a student come in to my lab one day. She was enrolled in my class. And the class was called Revenge of the Killer Microbes, which is about as fun as it sounds. It's a really cool class. And she comes in and says, Dr. Van Laar, I really like bacteria. I said, cool, me too. And she was a great student. So I was like, yeah, let's, I want to do undergraduate research. Great. Okay, but I also like birds. And I was like, oh my God, me too. I love birds. She goes, okay, but I don't want to do like only bacteria. I want to do bacteria and birds. And I was like, oh no, because I didn't know how to do that. So we talked to the bird guy. We figured out some ideas and we presented them to her. And she was like, no, I don't like that. She was like, I like one type of bird. I said, okay, well, what type of bird is that? She said, crows. I said, crows. I was like, you're kind of creepy. Crows are really smart. They do recognize faces. They do hold grudges. They do seek revenge. I was like, I kind of looked at her and I was like, I don't know about you anymore, but I still like you. We'll figure it out. So what we decided to try to do was answer some questions about bacteria that live in crows. Okay, but like I said, I didn't have the money to pay somebody to do analysis for me. So I had to learn how to do bioinformatics because I had to teach my student so she could do the analysis for her thesis. So how do you learn bioinformatics? Well, you could take a class. And those of you who are here next spring, hopefully you will. I'll be teaching a class in bioinformatics. I would love to see you in there. I didn't have a class in bioinformatics. I had one in computational biology where we learned how to use websites like BLAST to search for gene homology and things like that. But that wouldn't help me answer my questions. So the other way you can learn bioinformatics is the route I took, sheer stubbornness and willing to teach yourself. So I don't know about my colleagues, but I imagine they feel the same way. We never want to stop learning. That's why we wanted to go into academia. My mom always said that if I could get paid to go to school for the rest of my life, that would probably be the best job I could ever have. Look, Mom, look at me now. I get paid to go to school every day for the rest of my life. It's pretty awesome. The challenge with bioinformatics, especially when you don't have anybody to guide you, which is why I'm going to offer a class, I don't want to leave you hanging, you fail a lot. You get used to your computer telling you, nope. If it's being polite, it'll say no. Okay, so you learn how to fail productively, but you also have to be willing to put yourself out there. See what other people are doing. How do other people answer similar questions to the one you have? I call it my Google foo. I'm very good at using Google, okay, to find those answers. Learn how to seek out resources. A lot of bioinformatics is the ability to find those resources that are available for you on the internet. It's almost certain that a program exists that can probably do what you want. You just have to find it and figure out how to use it. And there's huge communities available of people who are really, really good at this stuff and are happy to help you. So I went to that student. Her name was Lee. I said, okay, Lee, let's do this. What do you want to do? And in collaboration with one of the other faculty at Fresno State, Dr. Cotty, we decided to figure out if the microbiome was different between rural crows and urban crows. And we had a hypothesis that it probably would be. It's hard to find crows in what are called rural environments. And so we actually used crows that were in a rehab facility that was rural. outside of Fresno. They had they had a consistent diet of dog food and hard-boiled eggs. So unlike the crows that we would have on the Fresno State campus that were like, hey, is that a McDonald's bag? These, we at least knew what they were eating. So the urban crow kind of mimics more what a crow would actually get. The rural, not as much. So, some of the common tools that you might need to do bioinformatics include learning languages like R or Python, or understanding software management programs like Conda. And again, if I can do it, anybody can. Okay, so we did a couple of different things. Lee did a couple of different things when looking at the data from the crows. So one really cool thing that I think it's cool, it's also kind of creepy, that crows do, especially during the winter, is they come in to roost, and they can come in the thousands. So in Fresno, one of the roosts is directly outside of Community Regional Medical Center. It's the hospital in downtown Fresno. In Davis, one of the other sites where the crows like to roost is on the campus of UC Davis. Especially right along one of the roads. You can tell whose car's been left there a couple of days. It's not pretty. So what we found, this is it's a Bray-Curtis distance or dissimilarity analysis, where we looked at the community composition of, it's the fecal microbiome. So Lee collected crow feces, extracted the DNA, and we sent those for sequencing of the 16S rRNA gene. And then we had to learn to use a couple of software programs to analyze that. And so in the green, or bluish green, these are our urban samples. The squares are from Fresno outside of the hospital. The triangles are from the UC Davis campus. And then these are from that, we called it rural, but it's from the rehab facility. So we saw that urban and rural crows do have a different microbiome, and that was pretty cool. One of the other things we did that we were interested in is, are the bacteria that are found, especially outside of the hospital, do the bacteria in the feces harbor antibiotic resistance genes? And we published a genome announcement where we sequenced a strain of bacillus, and we found that it was multi-drug resistant. Both we confirmed that in the laboratory doing laboratory testing, and also computationally using the whole genome sequence. What was actually really cool / sort of terrifying is that we also got some of the data from the infectious disease physician at the hospital, and a lot of the bacteria that we were seeing in the crow feces was also what they were seeing in their patients. And again, I'm talking like 50 feet outside the entrance to the hospital, so people are just walking through this poop and walking right into the hospital. And this was, of course, pre-COVID, so nobody's wearing masks or anything like that. And this is a video that Lee took of the crows coming in to roost at UC Davis. They just keep coming. So I think it's cool. I lived in South Texas for 8 years. We don't really have crows, but we have grackles that do a very similar thing. They're a little bit smaller, and they still make all those loud noises. Okay. What we also found, just computationally, was that overall, the whole bacterial community, we did what's called metagenomic sequencing. So we didn't just sequence only that 16S rRNA, we sequenced all of the bacterial DNA that was found in those feces. And then we did a computational analysis to find that we don't know which species had it, but overall there were species that had resistance to aminoglycosides, bacitracin, beta-lactams, chloramphenicol, fosfomycin, you can read the rest. So there was a lot of antibiotic resistance in that community. And when you're thinking about bacteria, that can be really scary because in a community, bacteria have a tendency to do what? Those of you who've taken bacteriology, what do bacteria do? They share DNA. Right? Conjugation, transformation. Right? So if one bacterium has antibiotic resistance, what's to stop the whole community from eventually picking it up? So those are kind of the implications. This research, I showed you the publication. We also had 2 presentations at national conferences. Lee presented this work at SACNAS and not ABRCAMS. I don't know why I put ABRCAMS. At the AOS, the American Ornithological Society meeting. So that was really fun. And we learned a lot of cool software tools to do that. And so one of the things that I want you to understand about why I like working in the CSU system is that the science you do, or the science at least that I like to do, is not always the same. So like some faculty have one project and they're super passionate about it and they make tons and tons of progress on it. And then sometimes a student comes in and says, I'm actually kind of interested in this. And you're like, yeah, that sounds really cool, let's do it. Or a colleague might come by and say, hey, I'm interested in this. What do you think? Okay, let's do it. So there is a species of bird called a bush tit, and they're very little. They're very little. They're so cute. And they make these weird nests. So normally when you think of a bird nest, you probably picture something like a little bowl, right, in a tree, put their egg in there, and then they sit on it. But bush tits make these weird nests that have a big opening, well, not a huge opening, but a little opening, because they're little birds, at the top, and then it hangs down, and inside is where they lay their eggs. And sometimes the parents and sometimes unrelated individuals will all come in, and they'll all sleep in there in the night, and I just picture this little tiny, nest full of these little fluffy birds. That makes me happy because it seems like it's so cute. But this nest was hanging right outside of our building, and then facilities came through and did what facilities does and chopped the branches. And so my colleague, he goes, I wonder if there's a difference between the opening and the nest part of it, where they sleep. And I said, well, we could do that. It'd be another microbiome analysis. So do the openings of the nest have different microbiomes from the nest area, from the brood area? And these birds are actually super prolific. They nest all over the campus at Fresno State. I imagine they do here too. I just haven't ever gone out to look. But they were right outside our building. We ended up finding 6 or 7 nests one year that facilities was always taking down. And so we did that microbiome analysis, and we did another one of these Bray-Curtis dissimilarities. And what we found was a little bit contrary to our hypothesis. We thought that the opening, because it's exposed to all that air and the birds coming in and out, the parents will take the feces out of the nest. There's a lot of traffic in and out of the nest. So we thought that that opening would be different from that brood area. And what we actually found was that each nest itself was different from all of the other nests. So the opening and the bottom were not that different, but all of the different nests we found were different from one another, which when we thought about it later kind of makes sense because all of the birds are different. But it's one of those things where just because your data don't support your hypothesis, it doesn't mean your data are wrong. And for me, when I was a very young scientist, that was always hard for me. I'm like, well, I thought this was supposed to happen, and this did not happen, so I must have done something wrong. No, the data are the data, and that's it. We also found one of the nests that facilities took was one that the birds had started really late in the year, and by July, we had stopped seeing activity, and the parents had abandoned it with the dead nestlings in it. So there were like 3 or 4 dead nestlings, and my student was like, well, can I try to grow some bacteria from there? I said, sure. And she wanted to see if they were drug resistant, and they were. So Enterococcus faecalis is related to the Streptococcus bacteria. They do cause human infection. And so we found a drug-resistant strain of that in the nest. And it's kind of hard to describe if you've not actually been to the campus, but there's a giant eucalyptus tree outside one of the science buildings. And this particular nest, I mean, I'm 6 feet tall. I've got a decent reach. If I were jumping, I'm not going to, but if I jumped, I could have touched the nest. So that's how close it was to the students as the birds were dropping the feces out of it. Okay, so these were really fun and just kind of little projects where a student was interested or a colleague was interested. I'm a microbiologist. You talk to me about bacteria, I'll sit and talk to you all day. So I was interested, but then I became the bioinformatics go-to person. And I'm like, well, you know, whatever, this is fine. So somehow I got hooked up with ag, and we were doing chicken genome analysis. I got hooked up with the plant people, of all people. Plant people. No offense, I'm just kidding. Like, these 2 faculty are, they're also some of my favorite people, so I'm just joking. And we did even some microbiome analysis of plants, and specifically sunflowers. So there's this parasitic plant called dodder, and it parasitizes sunflowers. And John Constable is a sunflower guy. Like, he found this type of sunflower that has a woody stem. So it's like a perennial instead of an annual. He loves sunflowers. So he found a population of these sunflowers that were parasitized by this other plant called dodder, and they wondered, are the microbial communities different? And so we published a very small paper showing, so this is the parasite itself. So as you might imagine, the bacteria in the parasitic plant, they all were fairly similar. And then we have the bacteria in the unparasitized sunflower that kind of cluster over here, and then in the parasitized sunflower over here. And so we found that the parasite microbiome was significantly different from the sunflower microbiome. But we also found some significant differences between the parasitized and unparasitized plants. So that was kind of a cool thing. I had never done anything with plants before, except for I did take botany here as an undergrad. That was about it. That was my last plant experience. So it was really fun to kind of be the bioinformatics go-to. And then the lizard people found me. I'm okay, whatever kind of bacteria you want, it's fine. So there was a faculty member at Fresno State, Dr. Telemico. He has since taken a position as the director of conservation at the Fresno Chaffee Zoo. If you've never been to the Fresno Chaffee Zoo, go, it's awesome. So he was interested in these western fence lizards and how when they try to raise them in captivity, the hatchlings have very poor survival. And he thought, well, maybe the microbiome has something to do with it. And my lab was downstairs, and so he thought, well, you know, do you want to, do a little microbiome stuff? And I said, sure. So we were looking at whether or not exposure to either adult feces or natural substrate or both could affect the microbiome of the juvenile and maybe make changes to their survival, improve their survival. So hatchlings were placed in 4 treatment groups. One was like standard, the control, nothing added. One that they got natural substrate. So from where the adults were captured, they took some soil and used that as natural substrate. One where they had no substrate, but they were exposed to adult feces. And then one group where they had the feces and the natural substrate. And one of the first trends that we saw was that in the juveniles that were treated with both, they got the neat soil from the normal environment, and they got the exposure to the feces. This is a metric called alpha diversity. Those of you, have you taken ecology yet? Yes, do you know alpha diversity? Dr. Gerson is like, you better if you were in my class. Anybody want to give me a... It's okay, you don't have to, I get it. It's a big class. How about Dr. Gerson? Alpha diversity?
You can define it for the way you use it.
Okay.
You go ahead.
Just very simply, and again, this is coming from a microbiologist. I am not an ecologist, so I will say that. It's a metric of how many species are found in a community and how many members of those species are found in a community. So the richness of a community, how diverse is that community, and abundance. Well, some metrics take into account both. Some take into account only one. And so what we found was that by that metric, by looking at the alpha diversity, the richness or the abundance of certain species, we found that the adults and the double-treated juveniles were relatively similar, but they were very different from the untreated or the single-treated juveniles. And again, the sample sizes aren't huge because they have really poor survival, so they didn't have a ton of juveniles for us to work with. So that kind of gave us an idea that maybe exposure to the adult feces and the natural substrate does make a difference in the microbiome. And we saw something similar when we looked at the community composition. So not only are we interested in, the number of species that are found, but also are those species the same? So are the species found in me the same as the species that are found in you? How different are they? Where do we differ? If I'm looking at my dog Barney, how different is that? Right? So we look at the community composition as well. What bacteria are there? Not just how many. And so again, what we found, I'm not going to go into all the details of this graph, but we found that the adults and the double-treated lizards, the double-treated babies, had similar community compositions. And they were very, very different from that completely untreated group. Okay, so I was like, yay, my job's done. I just had to tell them the microbiology. But I did want to know, did it make a difference? In terms of survival, not really. But they did find that the body condition was a little bit better in those hatchlings, including mass as one of their metrics. And so we're working on trying to write this up right now, but we'll see. Now that Dr. Talamico works at the zoo, it's kind of hard to pin him down. I'm going to skip this story because I want to tell you a little bit about something that is in progress instead of something that's already finished. So in progress, maybe what you've kind of noticed in this transition through this whole seminar is, first I just wanted to learn what was there. And a lot of early, especially microbiome studies of the bacterial community on or in a host, a lot of those studies right at first were really what is there? And you could get that published and everybody was like, oh, you're so great. You told us all the bacteria that are there. But unfortunately, you can't just get that published now anymore. And that's not as interesting as, sure, what's there, but what's it doing? So now we try to find out what's happening. And so the current project I'm working on is with my friend, Dr. Slade from Fresno State. And this is what kind of led us to this project. In late 2020, early 2021, there was a salmonella outbreak, Salmonella typhimurium, linked to a particular species of wild songbird called a pine siskin. There were 29 illnesses, 14 hospitalizations in 12 states. Of course, you see us there in blue. We had the most with 8 patients. And so that was very interesting. And then, of course, I'm smart, but not always smart. So I saw this little bird in my backyard one day laying on the ground, and I thought, ooh, that's not good. So I picked it up. Never pick up a bird that looks sick, ever. It's not a smart thing. I told myself I'm doing good because my chickens will eat it if I leave it down there. Yeah, I wanted to pick it up. So I picked it up, and it died pretty quickly. It was very, very sick. So I was like, I'll take it to work. I knew about the salmonella outbreak. I knew it was pine siskins. This is a pine siskin, by the way. I took it to work. We swabbed the cloaca, streaked it on salmonella isolation auger, identified salmonella in this bird. So we pretty much decided that this bird very likely died of salmonella infection. It had all the symptoms. And so when we did the whole genome sequencing and we looked for antibiotic resistance genes, we found a lot. So this is the video of the results. You can see there's just like 26 different antibiotic resistance genes in the genome of one salmonella isolate, which has pretty big implications if people were getting sick with this type of bacteria. Okay? So now we were looking at this in other species of finches. Why did only the siskins die? Was basically our question. So there's a subfamily of finches, very common in the valley. If you have bird feeders, especially if you have thistle feeders, you'll probably see them. But you have pine siskins on the top, you have lesser goldfinches in the middle, house finches down at the bottom. And pine siskins and lesser goldfinches are in the same genus. They're pretty closely related in terms of taxonomy. They're right there in the same genus. But why do only the siskins die? So Dr. Slade is an eco-immunologist. He studies the evolution and ecology of the immune system in non-human animals. And so he and his student performed bacterial killing assays. They took blood from siskins, from goldfinches, from house finches, and exposed a culture of salmonella to the blood. And the idea would be if the blood is good at killing the bacteria, that might be why the bird is resistant to dying. So the hypothesis was that maybe goldfinches and house finches have something in their blood that helps them kill the bacteria, whereas siskins don't. So it could be complement, natural antibodies, lysozyme, phagocytic cells, but the preliminary results suggested that only house finches are able to clear the infection through this mechanism. The bacterial killing assays, both siskins and goldfinches were bad at it. But still, the goldfinches don't die. So now what do? Well, we don't ever do this, but when we found out a grant was closing on June 30th, and so June 15th, I texted Joel and I was like, yo, what are you doing for the next couple of weeks? And he was like, why? I was write a grant. And he was like, yeah, okay. So we spent a couple of weeks writing a grant. And Dr. Slade is an immunologist, right? So he is interested in looking at more killing assays, immune cell ratios, and immunogenetics, looking at the MHC and toll-like receptor diversity in all three species. I'm a microbiologist, and so I'm going to be looking at what bacteria are found in these different birds. Do those bacteria harbor antibiotic resistance genes, and which bacteria harbor those antibiotic resistance genes? And so we found out relatively recently that we were awarded $467,000 from the NSF to study this question. So we're very excited about that. I also got a $10,000 RSCA grant from Stan State to continue doing this work. So we're really excited about our continued collaboration. It kind of looks like I don't know what I want to work on, but because I want to work on everything. I have lots of things I'm interested in studying. I have lots of opportunities for students who are interested in learning these techniques. These are some examples of just the bioinformatics-based projects I've been working on. Evolution of antibiotic resistance in Klebsiella pneumoniae biofilms. Effective litter amendments on the microbiome of broiler chickens. Another chicken person found me. These are two separate chicken people, which has been a really cool project with Fresno State. Transcriptomics of toxin-antitoxin mutants in Xylella fastidiosa. This is a bacteria that causes a disease called Pierce's disease in grapes. And that's in collaboration with Lindsay Burbank from the USDA, who will come and give a seminar later this semester. I have another project with Dr. Slade where we're trying to correlate the immune response, the microbiome, and malaria infection in white-crowned sparrows. And then I have two that are cure-based projects. One is just taking off where the Joint Genome Institute has like 2,000 genomes that are sequenced that they call orphaned. The PIs don't seem to care about them anymore. And so with faculty at Chico, San Francisco, Monterey Bay, San Marcos, and Santa Clara University, which is not a CSU, but that's okay, nobody's perfect. We're gonna try to come up with ways that we can have our students do bioinformatics analyses to get these genomes published. And finally, those of you who have taken or are taking genetic biotechnology with me, you know about the Pathways project where we're annotating Drosophila genomes. And again, each of those annotations should lead to a publication for the students involved. And of course, as always, which is gonna be what gets me in trouble, I'm open to ideas. Like, what else do you want to talk about? What else do you want to work on? I'll do it. I love bacteria. I love bioinformatics, even though sometimes it's difficult and makes me sad. But at the end of the day, it always makes me very, very happy. So I'm happy to take any questions.